Hi, everyone. Today's lesson is on the human genome and manipulating the genome, a really cool sort of application of some of the genetics we've been learning about. I think it's really the future of medicine as well. Many of you have shared that you want to look into a career in medicine. So we're sort of meeting together now the genetics part of it with that future sort of treatment idea. So I think one of the most interesting lessons that we do in this unit and maybe even in this course, I have included some links to videos which I recommend that you watch. Really, as I was saying before, there's a lot of ethical concerns that go into a lot of these things. And so currently, where there might not be policy on something, I think it's important that we look at both sides, both points of view. So I will be encouraging you to do that with a couple of videos in here. They're not mandatory to watch, but I would highly recommend them if you do have the time. So I want to start off by just telling you a little bit about the Human Genome Project, and then we'll sort of define what a genome actually is. So Human Genome Project, it took about 10, 11 years to complete. And it took many scientists to put together information from around the world. So what their goal was to do was find the sequence of A, T, Cs, and Gs, which make up the human chromosomes. And they have done this since with other organisms as well. So you can just simply search up the genome of different organisms. For humans, we're going to talk about its 3 billion base pairs. So that's a very huge genome. So I was mentioning before part of why the chromosomes are so condensed and why they have to be coiled around histones and so forth is that there's so much information stored in there that we cannot possibly use it all at one time. So more about that will come in grade 12. But really what I wanted to just mention right here was what their goal was to do was find that sequence of bases. So if you look from one individual human to another, we are something like 99.9% the same. So the sequences of ATCG are alike. That tiny 0.1% of bases that are different is what gives us our different physical characteristics. So I often get the question about different races. So human races, how different are we genetically? Well, even people within the same, what we call a race, we are so similar genetically that races are actually a human construct. That's not a construct of biology. And so, as I said, there's really only 0.1% that is different about any of us. So they have found that sequence. And I'm going to talk about in a few minutes, like what do we do with that sequence? Because that's where it gets really interesting. That's where we get into things like personalized medicine and treatment plans and all of that kind of stuff. So we had a couple of different people, or I should say groups, who were working to sequence the human genome. As I said, it took a long time. So they started in 1990. Around 2000, they eventually came to figure it out. So it took about 11 years, as I said. There was a private company as well as a public group of scientists working to do that. And of course, in the private sector, those companies are aiming to sell the information in some way and can make lots of money off of doing so. Now, today, we've realized because obviously our technology has improved, we're now in 2020, so over the past between 20 and 30 years, we've been able to really look at the specific human genome. So we've zoomed in on those base pairs, those A, T, C, and G. So what we've found is that we have coding sections and non-coding sections. The coding sections of the DNA are the sequences for genes that produce a specific protein. So those would lead to some phenotype or some physical attribute. So we spoke about in Mendelian genetics, it can be something simple like the color of something. So only about 2% of our entire genome, which is 3 billion base pairs, only about 2% of that, a really tiny proportion, is actually coding for something. So each individual gene has its specific protein that it codes for, and those proteins go on to do lots of different work in our bodies. More about that will come in grade 12 biology if you decide to take it, where we essentially talk about what's called the central dogma. I'll introduce it to you in the next lesson, actually mutations lesson, which is that each gene is responsible for a very very specific protein sequence. And so if there's anything amiss or mistaken in that gene, it will also be a mistake in the protein a lot of the times. So that's coding DNA. So all the alleles we were talking about, all those specific genes, those all fit under coding. And then we have the non-coding section, so the other 98% of the base pairs. And I put here in brackets junk DNA. In fact, it's not really junk because we've found out since that it's actually extremely important for things like the structure of the chromosome. It's also what forms the centromeres, which we talked about in meiosis, which allows the chromosomes to be separated and moved around and all types of things like that. They do not code for specific proteins. So they don't contain information for proteins. They are more structural. I mentioned here also this term telomeres. Telomeres represent the ends of a chromosome, and I'm going to link up a video about telomeres just for interest's sake. Telomeres are extremely important to prevent damage to the length of the chromosome. And again, we learn more about that in grade 12 biology, so I'm not going to really go into how important they are, but we are finding, and more research is being done, there's a connection between telomeres and cancer, as well as telomeres and aging. So if we can sort of crack the secret code of telomeres, we may be able to solve some issues surrounding that. So that's just a bit of information. So again, it's called junk DNA just because it doesn't code for a protein, but it's still extremely, extremely important. So other genomes I've just mentioned here, the number of genes is not necessarily proportionate to genome size. So we have 3 billion base pairs. We have between 20 and 25,000 genes. It's possible that some organism has far fewer base pairs, but has more genes there. It just means their genes are shorter or longer than ours. So that's all I mean by that statement. Now I want to talk about what do we actually use this genome for? And it's called functional genomics. So we have this genetic code. And now it's the question of what do we actually use it for? So what is like the power of knowing the human genome? And so I've mentioned a couple of things here. Functional genomics currently is trying to discover the function and importance of each gene. As I mentioned, there's thought to be between 20 and 25,000 genes. There's a lot of work to be done figuring out what each of them does. And so that is a huge, huge area of research. Also, I was mentioning how we treat genetic disorders. So we're going to talk about really soon is gene therapy, as well as genetic engineering. So we've sort of been able to crack the code and use our genes to our advantage in some scenarios. So more about that soon. Another place where you might have heard of it being done are things like DNA fingerprints. We know that we all have unique fingerprints. So too is our DNA unique. So even though it's only like 0.1% different from each other, that's still different enough that we may be able to do some DNA analysis. So the picture that I've put on the bottom right is basically what you would use it for in like a crime scene type of setting. That I can collect a DNA sample from a location and I can compare that against the DNA of other people. Now, I'm not going to go through the details of what this is. It's called gel electrophoresis. You learn all about it next year in grade 12 biology, but essentially we are separating fragments of DNA by size. And so what we're looking for are similarities and how close the size relates between like a location such as a crime scene and a suspect. A similar thing would be used for something like a paternity test. So comparing sequences between two individuals can also be a use for this. Now, I want to talk a bit about genetic engineering. And so what this is making changes to the genome of an organism. So I mentioned earlier, which you may or may not remember, viruses are actually pretty useful to us. And we're going to see that genetic engineering and gene editing uses viruses to an advantage, actually. But the goal of any genetic engineering, just in general, is to produce recombinant DNA. So DNA that has information from more than one individual. So I'm going to show you a really well-studied and really familiar example, which is how we use genetic engineering to make insulin. There's two sort of like most basic tools that we need to do this. One is the plasmid, which we spoke about in our bacteria lessons. The plasmid is a small circular piece of DNA that is in bacterial cells. And secondly, we have what are called restriction enzymes. You might also hear them called restriction endonucleases. And their job is to cut DNA at very specific points. So the goal in doing this is that I can take a piece of human DNA and insert it into a plasmid. So what I'm showing down here, and again, in grade 12, you go into way more details about this. So we're only doing an introductory example now, is I can actually take a gene for human insulin, I can put it into a plasmid, and I can essentially utilize the bacteria to make lots and lots of insulin for me. So what we're essentially doing here is cutting a very small sequence that is known from the human genome and pasting it into a bacterial plasmid. The goal of doing this is that we know that bacteria reproduce by binary fission, really quickly making exact copies of itself. So in doing that, what they're essentially doing is making lots of copies of this plasmid. And this plasmid, of course, as I said, has a human insulin gene inside of it. So unknowingly, these bacteria are reproducing exponentially, extremely, extremely quickly, and they're making human insulin for us. So at the end of the day, where we get all these cells that are now producing insulin, we sort of like tricked them into doing that. We then collect it and we remove it from the bacterial cells so that people who require insulin injections, they're getting human insulin, but it's actually produced by bacteria. So we've essentially manipulated that genome or engineered that genome of the bacteria to produce human insulin for us. And you may or may not know this, but before we knew any information about genetic engineering, they would actually have to extract animal insulin out of various livestock. They'd have to take out their livers and essentially grind them down, their pancreas, and grind them down essentially to get insulin that would then be injected into someone. So this method is extremely, extremely efficient. It allows us to produce lots of human insulin, and then that goes on to the pharmacy, right? That goes on to people who need that for type 1 diabetes. So moving on from there, so that's like a really basic genetic engineering technique. It's very well studied. We've done it for a long time. We're very familiar with it. Now we're coming into something called gene therapy, and we haven't perfected gene therapy as yet, but we have found lots of positive sort of outcomes from gene therapy. And what this essentially is taking away an unwanted gene in a genome and replacing it with a more desirable gene. So if we're thinking about people who have genetic conditions that are based on just one single gene, we're able to actually go into their genome and edit that out and put in a proper copy of the gene. So we do make use of viruses for this. And I had mentioned way long ago, Unit 1, that viruses actually do have a useful purpose. You might remember that viruses' whole goal is just to get into human host cells and insert their own genetic material. So what we essentially do here is we, again, almost like trick or manipulate the virus. We put a human gene of interest into the virus, and then we insert the virus into the person. We actually inject them with the virus. And our goal is we're getting the virus to infect a target cell. So I put an example here, the virus that causes hepatitis, its whole job is to infect liver cells, okay, in just a general term. So if we wanted our vector to insert a cell into specifically the liver, we could use hepatitis as a vector. So here's just like the basic steps. The first one is to choose a virus to infect specific cells. So if my goal was, for example, to infect the liver, I would choose hepatitis because that already infects that type of cell. Then I would modify the virus. So I would make the virus less harmful by removing certain components of it. And I would also put in the human gene that I eventually want it to input into the person's genome. Then I will infect the patient, which I know sounds kind of strange, but essentially we inject them with these modified viruses. And the goal is that these viruses will go around the body, removing those bad copies and inserting the good copies. So as it sounds, there are some issues with this. This is not a 100% perfect method yet, but it's getting better and better. So part of those videos that I'm asking you to watch is actually walking you through some really cool examples of gene therapy that they're actually doing tests on now, clinical trials on. So I would highly recommend those videos. Just want to go over why we say this is still experimental. We have had lots of successes, which I've given you some links down here. These are just within the past year or two. You can click into these links and they'll take you to various different articles and examples of where gene therapy has been useful. But here's some kind of problems with it. The virus might still be infectious. And so by putting it into a person, even though our goal is to modify a gene, we could actually make the person more sick. So that is an issue that scientists are trying to work out. How do we make sure that the virus does not cause illness, but does its intended goal? Secondly, is it's possible that the virus infects the wrong cell or it puts the gene into the wrong place. So remembering that there are 46 chromosomes in our DNA, in our nucleus rather. And so what if it's supposed to insert it in chromosome number 1, but accidentally does it in chromosome number 8? Well, that's going to disrupt the functioning of chromosome number 8. So you actually haven't solved the problem in doing that. So that is still a big issue that's happening. If the gene is only inserted in a somatic or a body cell, it won't be passed on to the offspring. So only genes that are edited in gamete cells, such as the gametophytes, we call them, early egg and early sperm cells, unless that's the location, then it's not going to be passed on to offspring. Okay. Now, part of what you are watching and reading about, which I've linked into right here. So here's our first video. Part of what you're doing here is looking at the technology. And the technology that we use for this now in 2020 is called CRISPR-Cas9. So what I'd really like you to do is to watch this video thinking about what technology they're using. It'll explain to you that CRISPR-Cas9 is essentially the tool used to locate and cut out different sequences. So you will see that as you watch the video. So this is just a little organizer for you to organize your thoughts on it. And then down here, after the video for genetic revolution. So again, they're going to talk a lot about CRISPR-Cas9 in here. And I want you just thinking about something that's interesting in there. You're going to find that they talk about other organisms as well, not just humans. But again, coming back around to all of this is what are the potential dangers and what are the ethical concerns? So most of us will watch those videos and find it uncomfortable. Because they talk about a lot of issues that we may or may not agree with. So I want you just to keep in your mind as we're looking at these questions for sort of benefits versus challenges, right? What are the concerns and then what are the positive outcomes? Ultimately, to do that, it's really to place yourself on the scale and express your own opinion about it. But in addition to that, you want to understand what the both arguments are so that if you did have a conversation about this or if you were faced with doing this, you can make an educated choice on it.